Hello, my name is Manuel Chavez. I'm a second year PhD student in Dr. Kara Butey's lab here at UTA, and I'm very excited to share with you my work on mycobacterial growth. So, mycobacteria are a group of rod-shaped bacteria that exhibit this polar growth mode, where peptidoglycan is added to the ends or the poles of the cell. Other rod-shaped bacteria, like E. coli, are lateral growers, where peptidoglycan is added uniformly. So we know that mycobacteria are polar growers, but we don't know the exact molecular mechanisms of that process. What we do know is that this protein called WAC31 is involved in polar growth because it localizes to the poles, and because when you deplete this protein, the cells exhibit this bulgy-looking phenotype. So my project involves characterizing this protein a little bit more. Here I'm showing you the predicted structural model of the protein, and here we have this IDR region of the protein, or this intrinsically disordered region, which is an unstructured region of the protein, which has a high probability of being disordered. And we care about this because these IDR regions are often associated with the formation of this biomolecular condensates. And these condensates are these dynamic and regulatory membraneless compartments that often organize biochemical interactions, and they respond to various molecular signals, and they are very sensitive and often selective on the different molecular processes that they're involved in. So we think in part that polar growth in mycobacteria is involved or with condensate formation. So one of the very first things I did is that I took this WAG31 protein and I just simply looked at it under the microscope. And here I'm showing you those images. Under a favorable condition, WAG31 forms these distinct spherical shapes, these condensates. Under an unfavorable condition, that same protein does not form condensates, but it's still there. If I were to take that protein again and put it in a favorable condition, it reforms this condensate. So this reversibility shows that these are in fact condensates. Okay, so that led us to create this preliminary model and how condensates may be involved in polar growth. Under an unstarved condition, under a favorable condition, we think polar growth is active in mycobacteria because this WAC31 protein condensate is able to localize to the poles, and other proteins involved in polar growth are localizing with it. Under an unfavorable condition, say a starved condition, we think polar growth is inhibited because we think that the biophysical properties of the condensate change. Perhaps it becomes smaller, perhaps it disperses, and that causes other proteins to delocalize, in turn inhibiting polar growth. So that model was based off the microscopy data shown here again. So to continue testing these hypotheses in this model, I made several mutations of the IDR region. Some of these were very small mutations, some of them were very long, but in turn, I think that the idea was that removing these mutations will probably inhibit or prevent condensate formation in these cells. Here I'm showing you the different phenotypes of these mutations, where cell length and doubling time, there wasn't much of a difference in these mutants. But when it came to swap efficiency or the likeliness for the cell to take in that mutation, we see that the longer the mutation, the less likely the cell was able to take in that mutation, suggesting there was some sort of viability. The cells were less viable. Here I'm showing you microscopy images of these mutants with the wild type normal WAC31 cell, and MMPL3 GFPs uses a proxy for cell division. We see that the mutants are not really much different. We see fluorescence at the poles. We see cell division as we should even in the mutants. So there wasn't much of a difference there. What was interesting though is under a starved condition, remember that normal mycobacteria are not dividing under a starved condition. And that's shown here with that green fluorescent protein signal is very dim. We don't see any fluorescence here, but we do see it in the mutations in the septa in the middle and at the poles in both of these mutations. So this might suggest that there is some sort of misregulation under starvation in the mutants. Perhaps removing the IDR is affecting the way the bacteria respond to stress signals here. And that data is further quantified in this graph here, where we see a very strong signal. Okay. So overall, hopefully I've been able to show you through this work that WAG31 forms these very distinct condensates, and that these condensates have a role in polar growth in mycobacteria, and these perhaps are very sensitive or involved in the regulation of how bacteria respond to environmental signals. And more broadly, this work will give us broader insights again on how bacteria respond to environmental signals, and including potential treatments or insights into how antibiotics work on actively growing bacteria. And thank you very much for your time.